In Trends in Immunology, we present a review on shaping variation in the human immune system. This is Adrian Liston on behalf of the authors. There is enormous variation that is present in the human immune system. This variation can be visualized using an immunological landscape. Different points on this landscape indicate different statuses of the immune system. For example, individuals at one point on the landscape may be Th1 biased, a second point may indicate B cell bias, and a third point may indicate a monocyte bias. Several different studies have tried longitudinal analysis of multiple individuals and have found that individuals tend to occupy certain stability islands, with the variation being relatively minor over time over the course of months or years. During an infection, the immune system does change. However, for acute infections, after the initial change in the immune system, individuals appear to revert back to the original stability island. This has been observed in the cases of acute gastroenteritis and also in influenza vaccination. By contrast, during the chronic infection, for example, gaining a CMV infection, individuals appear to radically change their immune system, and without a cessation of the infection, individuals seem to settle down in new stability islands. This means that both elastic and inelastic models of the immune system can occur depending on the nature of an infection. In this review, we also cover the drivers of immunological variation. One of the key drivers appears to be genetics. Multiple different studies have tried to quantify the effect of genetic variation on immune system variation. The rough figures that they are achieving is that 20 to 40% of the variation in the immune system appears to be driven by genetics. Another strong factor which shapes the immune system is age. While there is stability within the immune system over the course of months to years, over the course of decades, individuals shift from a profoundly precursor naive immune system to one which is more Th1 biased. A third factor is that of the environment. This environmental effect is most easily illustrated during cohabitation events, where two individuals start living together and now start to show a immunological phenotype that is much closer to each other than the original starting point. The cause of this cohabitation effect is not well known, but it may indicate that multiple small factors such as infection, diet, and microbiome are all causing a convergence on the immunological status. In this review, we cover the current literature and also pose outstanding questions for the future. If you're interested in this topic, please read our review in Trends in Immunology.